Okay, I think you can hear me now. Okay, I'm presenting this talk on behalf of Barbara Langill, an ex-PhD student of mine who's now living in Canada. But before I start, I'd like to acknowledge that I'm on the land of the Kaurna people, the traditional custodians of the Adelaide Plains, and I pay my respects to elders past, present, and emerging. Okay, the topic of my talk is regressive evolution, and this is the loss of ancestral traits during evolution, and it comes in many different forms, from loss of teeth in birds, wings in many different insect species, tails in primates, but also the loss of eyes and pigment in subterranean animals. And it was really this, the loss of eyes and pigment in subterranean animals that caused Charles Darwin some problems, and he couldn't see that there was a natural selection kind of way of potentially developing those particular traits in cave animals. 150 years later, and there's still much debate about how regressive evolution occurs in cave animals. And there's theories based on neutral mutation and genetic drift, direct natural selection, such as energy saving, indirect natural selection, such as, say, pleiotropy in particular genes, which might enhance some particular characteristic of a cave animal like taste and causing the loss of eyes. And it's really through some evo-devo studies of the Mexican cave fish, Astyanax mexicanus, that it's really selection through energy saving or pleiotropy have become the popular theories in cave biology. Evidence for neutral evolutionary processes in eye genes has come from a variety of different sources, mostly focused on vertebrates, marsupial moles back in 1997, IRBP gene, and also loss of function mutations associated with opsin genes and other genes involved in vision. And there's really just been one study of an invertebrate, which was a study by Remco Leys on the cinnabar bipigment gene. So what we needed was a different model system to study this process. And one really nice system comes from this region of Western Australia, where there are these calcrete aquifers, shown here in black, bodies of water distributed along ancient paleo drainage systems. And many years ago now, Bill Humphreys from the Western Australian Museum and Stefan Eberhard discovered a very diverse stygofauna and troglofauna from these calcrete aquifers. But one of the most interesting groups was this, subterranean ditiscid beetle fauna. These are water beetles that incredibly diverse in that there's now 100 species known from about 46 different calcrete aquifers. And there are between 1 and 3 species in each calcrete, and they have a very interesting non-overlapping size variation. One other interesting aspect of them discovered by Carl Jones is that they're living permanently underwater, so they're respiring from the water in contrast to surface species. Remco Leys showed that most of these subterranean species evolved independently between about 8 and 1 million years ago from multiple different surface ancestors. But there's also these sympatric sister species that are shaded here, where their ancestor is most likely to have been a Stygobiont. So the aim of this, the research I'm presenting here, is to investigate the molecular evolution of different genes involved in eye function in subterranean diving beetles and compared to surface species, and really to look at the evidence for neutral evolution of these genes specifically involved in eye function. So the predictions for neutral evolution are that really mutations should occur in parallel in independently evolved subterranean species. So these are different mutations in the same suite of genes. They should only occur in genes specific to the regress trait. And we might predict that recently evolved subterranean species will have fewer of these loss of function mutations than older subterranean species, just due to the chance fixation of a mutation and genetic drift to fix it in the species. And with our sympatric sister species, we might actually find that they share loss of function mutations. So the methods that have been employed here was first to identify candidate phototransduction genes. And this was done by some very nice research by Simon Tierney, and 21 gene sequences were discovered using RNA-seq data from surface species and also some subterranean species. Now, these can be used to design biotinylated RNA baits, and then through targeted exon capture, shown here, the baits have hybridized to their region of complementarity, attached some streptavidin-coated magnetic beads, and then you can fish out the sequences using a magnet, purify them away, and sequence them on an Illumina platform. So we then conducted, or Barbara conducted, bioinformatic analyses involving assembling the sequences, concatenating exons, aligning, phylogenetic analyses to verify the orthology of genes, and then we searched for loss of function mutations. So these are mutations that might cause a deletion, but in particular, a deletion which might cause a frame shift and a change in the amino acid sequence and the introduction of perhaps a stop codon. We also did evolutionary analyses to just look at the rate of evolution of amino acid substitutions in these particular genes. So if you have one of these genes from a surface species, you expect to find a nice untranslated region, a methionine at the start of the gene. Here I've taken out the introns, but it's got a conserved amino acid sequence and a stop codon, and then another 3' untranslated region. But what we found in a bunch of different phototransduction genes was that they had a variety of different mutations. And in this particular case, it's an alignment of 3 subterranean sister species with a surface species. And what we found is an 18 base pair deletion and the introduction of a stop codon in the sequence a little bit later on. But in one of these species, it also has an independent mutation resulting in 11 base pair deletion, causing a frame shift, a change in the amino acid sequence. The stop codon goes back in frame and it has another stop codon further on. So these sorts of changes here resulted in 96 amino acids being lost from the protein. And it's evidence that these genes are evolving under relaxed selection. So Barbara carried out these analyses on 18 different phototransduction genes. So these are genes involved in the detection of light and the light detection pathway. And she obtained these sequences from about 31 subterranean species and 6 surface species. And this is a tree showing the relationships among the species, and I've highlighted sister species among them. But if you notice on the graph on the right, the green indicates a complete open reading frame for the genes. Yellow indicates an interrupted reading frame, so loss of function mutations occurring. And if we find in the case of the surface species that they show essentially open reading frames in all their genes. In the subterranean species, the vast majority have interrupted reading frames with a few exceptions. So INA D is a gene that is under purifying selection in these beetle species. 8 of the 18 genes were found to have mutations leading to loss of function in these particular genes. And these include arrestin genes, opsin genes, three different opsin genes, TRP channel genes, and a gene called NENA-C. Also, we found that in genes, some of these genes in recently evolved subterranean species like Limbodesus microocular at the top of the phylogeny, it actually shows no mutations in any of these particular genes. So that prediction that recently evolved species may show no evidence for loss of function mutations bears out. So all of these particular proteins are key elements of the phototransduction network, the opsin genes detecting the photons of light, the TRP channel genes in the inner C leading to the calcium ion channel response, and arrestin genes converting the rhodopsin back to its native form. So in discussion, convergent loss of vision in cave animals may well be explained by neutral evolutionary processes. So the fixation by genetic drift of random mutations under relaxed selection. The caveat to this is that we can't entirely rule out selection driving actually the loss of eyes in subterranean animals early on in their evolution, but I think the process is potentially sufficient to lead to the loss of vision over time. A major question really is how many genes actually evolve neutrally in these subterranean beetles compared to surface species. There are multiple different regressed traits like loss of eyes and pigment, wings, and also, as Perry showed, reduced thermal tolerance. What I think these beetles represent is what I would say is the Goldilocks temporal evolutionary time zone. It's coined by Simon Tierney, just right for detecting neutral evolutionary changes in these aphotic environments, but not too old that you'd actually lose these genes and fail to detect them in the genomes. I better finish there, and I'd like to acknowledge a variety of people who've contributed to this research over many years now, and also some funding partners as well who've contributed to the research costs. So thank you very much for listening.
Thanks, Steve. If you can unshare your screen.
If I can, there we go.
Do we have any questions for Steve? Steve, there's a question in the chat from Ari. Would you like me to read it out, or do you want to read it and then answer it?
Sure. Do you have any sense of the relative mutation rates in subterranean versus terrestrial environment, given the nature of the environmental conditions? Yeah, that's a good question, Ari. There's actually uranium in these calcretes, and so it's quite possible that there are faster evolutionary rates in these aquifer systems. And we don't have a handle on, at the moment, what those rates are, relatively speaking, but it would certainly be worthwhile exploring. Remco Leys has verified that there are sort of higher, well, he took a Geiger counter out to these aquifers and found very high levels of radiation. So that's confirming that it is potentially an issue. Okay, there's another question. I don't know if I've run out of time, not quite. How do you know that the frameshift mutations haven't been positively selected for? Yeah, that's a really good question. We've actually got transcriptome data from a few of these subterranean species and found that evidence for the continued expression of some of these genes. So there's a pseudogene expression, and I think that would be unlikely to occur if there was actually natural selection driving the loss, or sorry, helping to fix these frameshift mutations. We would have expected them to be switched off.
Okay, we might have to call it for the questions, but you can always ask Steve directly through the chat if you had a question that you